All right, welcome this morning to our lecture on the beginning the female reproductive system. We'll probably take, well, definitely Monday, Friday, Tuesday, Wednesday, Friday, Thursday, Friday. Well, definitely today and Monday on female reproduction. We'll see what we can get through. Before I started the video, I was showing those who are here that there will be a handout in the announcement. Nope, in the modules. So they're called on female reproductive cycles, because there are 3 reproductive cycles. There's an ovarian cycle, a uterine cycle, and a hormonal cycle. And you need to know what's going on every day in the quote unquote 28-day cycle that of course a man came up with, because no women have 28-day cycles unless you're on the pill. But no, more than not have irregular cycles, that we have weird stuff that goes on. Anyway, but I'll talk more about that. So make sure that you bring this to class on Monday. And Tuesday, I think, because we don't have lab on Tuesday, that I'm still going to go ahead and have a lecture at 9 o'clock on Tuesday, so we can keep moving along and get ready for our test on the 4th, because I still want to have the test on the test number 6 on the 4th, and then we have 5, 6, 7 to get ready for, or 5 and 6, so the 5th and the 6th to get ready for the final on the 7th. Still investigating some options on the final, but you will be where you're at to take the final. You won't be coming here or anything. So my University of Florida just went to a new thing, and so I've got to investigate that and see if that's something that we can do for our final. But anyway, you'll know next week in plenty of time. Okay, so let's just go ahead and sally forth and go on to our female reproductive system. And okay, so watch me while I drink my breakfast. All right. So when we were doing the male reproductive system, we talked about spermatogenesis and the fact that the spermatogonia, or the germ cells, the ones that are going to always go through meiosis, are going to give rise to the sperm. So that same process, well, the meiotic division is going to be the same, but the results are not going to be the same. In fact, oogenesis in a female is quite distinct from spermatogenesis in a male. And when it happens and what the result is. And so we're going to go through that right now to help you understand what we need to make in with oogenesis. So I'm just going to read through this, and then I'm going to go through the picture and in the next slide and talk to you some more about that. So during fetal development, the primary oocytes begin the first meiotic division. So as the ovaries are forming in a female, the oogonia that are going to come from the yolk sac that are going to give rise, they're going to go through meiosis and become ova, are actually going to start meiosis. And they're only going to do prophase 1, but they start in prophase 1. So all of the oogonia that are in the ovaries have begun to, they've replicated their DNA. The homologous chromosomes have found one another, and I'll show you what that looks like in a minute. And then they stop, and then they do not progress through the rest of meiosis until the female is in puberty, and then follicle stimulating hormone comes and starts acting on those oogonia and causes the continuation of meiosis. So they go to prophase 1, and so tetrads form and can lead to non-disjunction. So let me see. I don't have a picture. I'm going to go back to this picture here in a male, and so this. Let me find my little Okay, so this stage right here, prophase 1, is where the oocytes, well, they're not oocytes yet, they're where the oogonia, so the cells that are going to give rise to the ova. Oh, I didn't silence everybody. I didn't mute everybody. Can you guys mute whoever you are, and I'll mute y'all in a minute. So they're going to sit like this. Okay, so we find the chromosome number 1 from the mom, the chromosome number 1 from the dad, number 2 from the mom, number 2 from the dad, and they're going to hang out together for 14 years on average. Okay, well, it depends on whenever menarche is, when the female has her first reproductive or her first menstrual flow. They're going to sit like this. Now, that's fine. So what? Here's the situation. Is that when these tetrads form, number 1, crossing over is going to occur, and so that's how we get like dark-haired, blue-eyed people and blonde-haired, brown-eyed people, and new, different combinations. I was going to say strange combinations, new and unique combinations that haven't been seen before, where pieces of chromosomes break off one another and then reassemble, and so then you have chromosomes that have new genes on them, new sequence of genes that hadn't typically been found, or new alleles. I guess it shouldn't be sequence of genes because they're homologous chromosomes. So new alleles that haven't been combined together before. And so the problem with these chromosomes sitting together for such a long time is that when they actually go into metaphase and then into anaphase, where the chromosomes need to, these guys need to go this way and these need to go this way, sometimes they fail to separate. So then you'll get 2 and 1 going this way, and then you'll get 1 going this way. And then when you go to here, then you'll end up with a cell that has 1 in it, a cell that has nothing, and maybe a cell that has the 2 and the cell that has 1. So these are the cells that we want. These are the haploid cells. We don't want this one because that's got nothing in it. And we don't want this one because we have an extra copy of a chromosome. And you guys have all heard of that, and that's called trisomy. So there are different trisomies. The most common trisomy is trisomy 21. For some reason, the 21st chromosomes don't separate very well, and so that results in Down syndrome. Now, usually when we see Down syndrome, we see it in individuals, in women who are older when they become pregnant. I was 37 when I was pregnant with Amy, and my doctor, he said, well, you're now at risk pregnancy. And I said, yeah, I know. And just because of my age. And he says, we could do all this genetic testing that they, is standard procedure. He says, but it's not going to do any, this not going to teach us anything. Even if there was a problem, you're not going to do anything about it. And I said, no, I'm not going to do anything about it. It would just let me know, hey, you got a kid with Down syndrome, And so I didn't have any of those tests done. And I had a friend who, with her first pregnancy, she was about 38. And her doctor had said the same thing to her. And she goes, well, he had said, you're an at-risk pregnancy now. We should do all this testing. And she's all, you don't need to do any testing on me. We don't want to run the risk of me losing this baby. And then the other thing, she says, I don't care what you find, I'm still going to have this baby. I'm still going to carry it full term. And so he's all, oh, okay. And it was kind of interesting because he hadn't had a woman tell him that before. And so I was very happy. I was very proud of her to just say, oh yeah, you're just so good. Anyway, so there's that. So let's go back to, whoops, we went through a whole lot of stuff. Okay, so that's what leads to non-disjunction. So the chromosomes fail to separate, and then you can get some trisomy. All right, now, the oogonia are going to be found in follicles. And so they're like little pockets. Let's look at a picture of a follicle. Okay, so here's going to be the oocyte, the oogonium. So it's going to be sitting here in a little chamber called a follicle. And so every single one of them are going to be every single oogonium is going to be in a follicle. So if we go back here, by birth, we have 2 million oocytes, or 2 million primordial oocytes is what we call they're not even primary oocytes yet. They're just primordial follicles that have the potential to finish out meiosis and become an oocyte. So this 2 million number is going to decrease over the girl's development to puberty, because many of the follicles go through a process called atresia, where they just kind of disintegrate, and they don't get activated. And so just like we have the extra sperm get reabsorbed into the man's body, some of the follicles that go through atresia will never develop, and most of the follicles will go through atresia. But by puberty, we have 400,000 oocytes remain, and each month a small number are activated by follicle stimulating hormone, but only one oocyte out of the bunch is going to complete meiosis to produce a haploid ovum. And so what does that mean? Once a month, we're going to have a cluster out of this 400,000, and many of these 400,000 are going to go through atresia as well. And so if you think about, well, how many months are there in between puberty and menopause? And so if we take 40 years and we do 40 times 12, that's 480 months. So that's about how many follicles mature in a female, is 480. Okay, so so that's all her reproductive fecundity, but you can't have 480 children, obviously, because once you form one, now you got to wait 9 months before you can have another one. So anyway, okay, so here's what happens. I want to take a look at this picture. So we're going to start with, we're going to get to this one. So and this actually should be 4N, because we have duplicated, right? But the primary oocyte arrested in prophase 1, present at birth. So now the ovary becomes inactive during childhood. And then each month from puberty to menopause, then when the FSH acts on it, and one of these follicles becomes an ovum, then we finish meiosis 1. Then after the sperm fertilize the ovum, then we go into meiosis 2. And so the female is not going to go through meiosis until she absolutely has to. And so I think that's kind of funny. It's like, I'm not going to waste my time going through meiosis because I know that's not going to get fertilized. Hang on, I need to, okay, all right. Okay, so here's what's going to happen. So let's think about what happens in a male. Let me write on here. So the male is going to go through the first meiotic division, and then he's going to get a sperm here, well, a spermatid and a spermatid, and they're going to be diploid still, and then he's going to divide and then produce a spermatid and a spermatid and a spermatid and a spermatid. And every one of them is going to end up like this guy right here and have the ability to fertilize an ovum. However, in a female, if you look at how meiosis happens, when we start meiosis, we're going to split. Here's the nucleus. And so we're going to do that first meiotic division. And so what we do is we package half of the chromosomes in this little tiny cell-like thing called a polar body. Now, a polar body has nothing but the rejected nucleic acid that we don't want. So 46 chromosomes are going to be in here. There's going to be 46 chromosomes over here in this cell, okay? And then as we finish, go through meiosis 2, once the ovum gets fertilized, then the polar body will finish. And so now we're going to get 23 chromosomes in this cell, and 23 in this cell, and 23 in this one, and 23 in this one. However, these cells can never be fertilized because, well, they can be fertilized all they want, but they'll never grow into a baby because there are no organelles inside of here. We put all the organelles into this one ovum. And that's why it's so big. And ova, you'll notice, are a lot bigger than sperm, because sperm don't have organelles either. Because they don't need to, other than mitochondria, they don't need to do anything but go swim and fertilize. And the female, though, when you realize that this ovum is going to get 23 chromosomes from the dad and now become a zygote, well, where's all the Golgi? Where's all the endoplasmic reticulum? Where's all the ribosomes? Where's all the everything so that cell division can take place in that new organism? Well, it's all cram-packed inside of that one ovum. And so we have this uneven cytokinesis going on, where all the organelles end up in only one ovum, and then we produce 3 non-viable polar bodies. So like I said, if a polar body ever got fertilized, nothing would grow from it, because there's no machinery to build a baby with. So that's why we have just 1 ovum in the female. So we get 4 for every meiotic division in a male, but we only get 1 mature ovum in a female. Okay, so I think that's all we need to talk about there. So we go to the next slide. Here's oogenesis. Then it results in 3 polar bodies and 1 mature ovum, which contains all the organelles and nutrients necessary for mitosis in the zygote. Okay, so does anybody have any questions on that before I go on? Okay. All right, so here is an ovum. Nope, here is an ovary with many ova developing. I want you to understand in this picture, even though it looks like the follicle is progressing through the cortex in a month, it's not. This just shows the various stages. And so they're showing what it's going to look like in 1 month for 1 follicle. But it's going to be in this spot. So let's say this is the one that FSH is acting on. It's going to go through all of these stages, but it's going to do it right there. So they just wanted to show the sequence of events, but not that it happens in a stepwise fashion. So, okay, so here's what's going to happen. You have thousands upon thousands of primordial follicles in your ovaries right now, girls, ladies, unless you're me. They're just sitting there waiting for something to happen. It's just like American Idol. So they go to the audition, and the FSH, so I don't know, Simon Cowell or whoever, I don't even know. No, he was never on American Idol. Was he? I don't even know. Anyway, the judges are follicle stimulating hormone, and they're going to pick a bunch, 20 or so, in fact, like the 24 that go to Hollywood. And so they're going to pick these, and they're going to start going through this, not this one, this process right here. So these primordial follicles in the presence of FSH are going to start changing the outside of the cell, and they're going to start building material around the outside that's going to prepare that follicle for ovulation. And so they're all going to work at it. They're all going to work at various levels based on FSH receptors and how much FSH actually bathes them, binds to their receptors. And what you're going to get is one that actually wins. So we're going to start out with the primary follicle, and then we're going to go, I'm just reading the notes for the next page to decide if I'm going to go there yet. Now we'll just look at this picture. Okay, so here are the 24 that went to Hollywood. So now they're going to start developing the outside, and they're going to grow 2 groups of cells. They're going to grow granulosa cells, and then they're going to grow another group of cells on the outside called the theca folliculi. So here is the theca folliculi on the outside, and then we have granulosa cells. I didn't even make granulosa cells. Oh, I probably should, but maybe next semester. So these progress, and as they progress into a secondary follicle, they're going to start to get a fluid-filled antrum inside of here. So a little pocket of fluid here that then is still going to keep progressing until one of them is going to get to this stage. And so this stage right here that is kind of like a zit that needs to be popped, this is called a vesicular follicle. And only one out of the 24 is going to become a vesicular follicle, because it's the one that's going to pop and ovulate, and out will come the ovum. Okay, so the other 23, they just didn't make it. They wait till another turn, or they go through atresia and they shrivel up and die. So the question is, what effect would cysts on the ovary have? So cysts on the ovary are going to be big fluid-filled cavities inside, and can interrupt that action of FSH on the ovary. And so therefore, if you have like polycystic ovarian disease, you may have a very difficult time getting pregnant, because that those cysts are interrupting the follicle's ability to develop, and so that's that. I lost my thought. Oh, so these guys then are gonna wait to see if maybe they get another chance at it, or like I said, atresia may happen, and then they don't get a chance to develop anymore. And so like this guy that went so far more than these other ones could have been from the round before that they went and auditioned again and already had a head start. So, but anyway, so FSH is going to be the hormone that stimulates this process up to this point. And then the process of ovulation is going to be due to the presence of LH, luteinizing hormone. So luteinizing hormone causes ovulation, and then hopefully this will get caught by the fimbriae and then brought into the uterine tubes, and then move down and hopefully encounter some sperm along the way. So once the ovum has now proceeded on its journey, what happens to the ovulated follicle? Well, it's going to turn into this yellow body right here called the corpus luteum. And the corpus luteum is a straight up hormone gland. Oh, I didn't tell you about the hormone gland. So here on the outside, the theca folliculi is what is going to produce estrogen in the female. And then once the corpus luteum has formed, it's going to produce both progesterone and estrogen. Here's a fully developed corpus luteum. It's going to last for 14 days. And then if nobody has moved into the uterus and taken up residence there, then it's going to degenerate and become this corpus albicans. All right, so let's go through the steps right here. So primordial follicles, those are the ones that got formed embryonically, and so they're just sitting around waiting for something to happen. And so we are in the follicular phase of the ovarian cycle. Okay, so that's going to start from day 1 to about day 14. Now, what does this day 1 mean? And I wish I had given you your charts. I wish I had been a little proactive and realized extent of when I was driving to work this morning, you didn't have it. What does day 1 mean? That is the first day of your menstrual cycle. Remember, this is a cycle. So just like with the cardiac cycle, we could start absolutely anywhere. We chose to start in mid to late diastole. We're choosing to start on the first day of your menstrual period because that's an event that we can see. So if we take a look at, well, what's going on in the day 1 of the menstrual cycle, that's a wrap up of the last cycle. So when it means that last cycle was unsuccessful, that fertilization did not occur, and so, you know, I know I've mentioned it to some class. Maybe I mentioned it to you. Maybe it was last semester, and I just don't remember. But I said your uterus is just like, you know, my kids are moving into my mom's house tomorrow. So I painted the room and I cleaned out all the stuff, and it's all ready for my son and his wife to move into her house. And so that's my, for lack of a better term, it's my fresh, clean uterus. And it's all set to go. So here my kids will be moving in, and they'll take up residence, and then I'll care for them. But let's say that nobody moves in, then what am I going to do? Just leave the room bare? No, I'm going to rip down the room and I'm going to build it back up again. And that's what your uterus does if nobody moves in. So since nobody moved in, on day 1 and we're ripping down to start construction again, now we're going to start advertising for a new tenant. And so that will be the development of the tenant, is what's going on in the follicle. Okay. I know that was a long or roundabout way of explaining that, but it's exactly what's going on. Okay, so what's happening then? Well, those primordial follicles are being activated by FSH, and the oocyte will enlarge and become the primary follicle. And then around the outside, and I'll show you a picture here in just a minute, it'll give you a little better idea where the theca folliculi is. The theca folliculus is actually, that's one. So the theca folliculi form to produce estrogen and a zona pellucida, which is a thick transparent membrane. And then the vesicular follicle will bulge from the surface by day 14. The primary oocyte at this point, if you're going to become ovulated, you finish meiosis 1 and you'll become a secondary oocyte, but only if you're the one that's going to be ovulated. Everybody else is going to stay in as a primary oocyte in metaphase, metaphase 1 or prophase 1. Okay, so let's take a look at this picture. This shows the stages more one after the other, more of a complete like cycle. So here's a primordial follicle right here. So all 400,000 are just like this. And now we're starting to get some differentiation of these cells on the outside of the follicle to become granulosa cells. And then we have these other cells around the outside that are also developing in the presence of FSH, called the theca folliculi. And so then as the FSH keeps stimulating and stimulating and stimulating this one follicle, then it's going to turn into develop an antrum. So this would be the vesicular follicle, and then the secondary oocyte inside of there. So there'll be one polar body in here. I guess that's what that's supposed to be. One polar body and the ovum. And then we're going to get LH being released, and then ovulation happening, and now we're at a secondary oocyte, and we've got these granulosa cells remain as the corona radiata here on the outside. Then we go to the corpus corpus luteum, which means yellow body, and then corpus albicans is white body. Okay, so yeah, it is yellow, it is white. All right. Okay, so ovulation then is going to occur near mid-cycle. So on a 28-day cycle, sure, it's going to be day 14 where ovulation happens. Okay, but let's say that you're an individual that has a longer cycle, and so we'll just use me as an example because I'm the guinea pig for everything. I used to have a 33-day cycle. So in my younger days, 33 days. So wow, when am I going to ovulate then? Do I ovulate 14 days from the start, or 14 days from the end, or do I do it halfway through, or whatever? And so how do we, hang on, somebody was trying to get in. I'm in Zoom. All right. So when is it? So we'll talk about that in just a second. Okay, so the ovary, the ovary wall is going to rupture and expel the secondary oocyte into peritoneal cavity, getting caught by the fimbriae, and one dominant follicle develops when the LH stimulus is given to ovulate. So that one follicle is just going to pop open. Fraternal twins will happen when more than one follicle ovulates at a time. So if you have identical twins, then you know it's because that inner cell mass divided, became an embryo, and then divided somewhere along to be two people out of the same genetic information. But in fraternal twins, the kids are no more genetically identical to one another than if they had been subsequent pregnancies instead of simultaneous pregnancies. So usually what happens is that you'll get one follicle developing in one ovary, another follicle develop in the other ovary in that FSH stimulus, and so then two ova ovulate at the same time, and then they will get fertilized by two completely different sperm. So it's just as if they, you know, they were 9 months apart. They're not going to be genetically more identical than one another. But that begs the question then, why do some fraternal twins look very similar to one another? Well, that has to do with what is the environment in which gene expression was occurring. So because those two babies were in the same, very similar, because not even babies will end up in the same environment, that one embryo will get more nutrients than the other one because of maybe umbilical cord issues and that kind of thing. But because they're in the same environment and they get the same nutrients at the same time, let me finish. The gene expression tends to be very, very similar. Okay, so there's that. All right, so the luteal phase. Okay, so now what's happening in the ovary? We've had the follicular phase, we've had ovulation. Now we're looking at the time in which the corpus luteum exists. So that's called the luteal phase. So this is corpus luteum activity from day 15 to day 28, and the luteal phase is always 14 days. So that now lets us know, and I'm going to go to my whiteboard so we can map some things out. Okay, so, okay, if we have a 28-day cycle, oops. So the follicular phase will be 13 days. And then ovulation, stop and think what it's called, ovulation would be day 14, and then luteal phase would be 14 days. Oh, well, okay, 14 days. So day 15 to day 28. Okay, but let's say that you have my cycle. So if I, whoops. I have a 33-day cycle. My follicular phase is 18 days long. Okay, so a full 5 days, because where's the extra 5 days? Because I go 5 days longer than someone with a 28-day cycle between periods. So that 5 days is at the beginning in the follicular phase. For some reason, it takes longer for my follicles to develop. Okay, then I ovulate on day 19 for me, but then my luteal phase was always 14 days, just like everybody else's. So then what if you have a 21-day cycle, like some of my students have had before? So now your follicular phase is super fast. Okay, so it's only 6 days. So it only takes 6 days for that follicle to develop and to ovulate. Ovulation, oops, is on day 7, and then their luteal phase is still 14 days. Doesn't matter who you are. Now, here's the funny thing about finding this out in myself. So I knew that I had a 33-day cycle, and because I'm such a planner that I like to plan out when I'm going to have my children. And so with Cameron, I had had 2 miscarriages, and then the doctor said, we were building a house and stress and everything. He goes, you know what, you guys, just relax. Don't worry about trying to get pregnant. And in fact, we didn't even live in the same house. I lived with my parents. He lived with his parents. And so it was just a weird, funky situation while we were building our house. And so just right after that, right after the doctor said, you know what, don't worry about it. And I got pregnant. And I was like counting days. And I was like, that's a little weird. When I realized that I was pregnant, when it would have, ovulated. And so anyway, then when it was time to have Missy, and so Cameron was like, I just, had got pregnant. And so he was born January 2nd, which is a really bad time to have a child when you're teaching school, is to have your baby in the middle of the school year. And I just, writing lesson plans, and I had to take a week of unpaid leave because they weren't going to extend my 4 weeks of maternity leave. And oh my gosh, anyway, it was a nightmare. And so I thought with Missy, I'm like, oh, it's now time to have another baby. And I don't want to have my baby during the school year. So I was checking months, to find out, well, I want my baby to be born in the summertime so I can be home. So I started trying to get pregnant. Nothing happened. Trying to get pregnant, nothing happened. I kept trying and trying each month. Nothing was working out. And I was always trying to hit that magical day 14 time, and even using the ovulation thermostat thingy, thermostat, taking your body temperature, which is dumb. It's such a pain. It doesn't work that well. And take it from me. And so I thought, okay, one more month. We'll try this last time. It was in November. I freaking got pregnant. When did I have Missy? August 26th. That was the absolute worst time to have a baby, because I went to school. Actually, my doctor said, hey, I do surgeries on Wednesdays. Do you want to have her this Wednesday? That would have been the 19th. That would have been 2 days after school started. I'm like, no, I got to be there. I had a student teacher coming in, and I had to get stuff ready for my long-term sub and everything. And I'm just like, let's just have her on the 26th. So I went to school that fall, that August, the 17th was first day of school, met my students, said, okay, guys, it's good to know you. I will see you in a month. And then I went and had my baby. So when I was going to get pregnant with Amy, I had just moved here. And I thought, I cannot have a baby in the middle of the school year. Can you imagine if I left you guys for a month and went and had a baby, and it's like this? It would be just a nightmare. So I said, I have to have Amy in the summertime. So I got an ovulation test kit. And the ovulation test kit taught me something that I didn't realize. Well, I kind of already knew, but didn't really already know. Which end is the 14 days. And so with my other children, I was going on day 14 was the day that I ovulated, thought my my luteal phase was the really long one. Well, come to find out, I was always trying to get pregnant 5 days before I even ovulated. So no wonder I couldn't get pregnant. So as soon as I realized with the ovulation test kit, what it does is measure your urine levels of LH, so that when those surge, then got to act on it right then, because there's a viable ovum. And so I did that in September, didn't hit it just right, didn't get pregnant, said I can do it one more time. And so I tested in October, got pregnant in October, had Amy in June. And so then I didn't have to be gone for a month. So if you want to plan when to, and such a planner, I've just got to be in control. And so yeah, I had Amy exactly when I wanted to have Amy. So if you're in that kind of a situation and you don't just, let things go to fate, no, fate's not going to control when I have children. Okay, so there's that story. I always have to tell that story.
Miss G, for the 21-day cycle, I took a picture, but I missed it. For the ovulation, it's how many days? Day what? 7.
Because remember, so 14 days is always at the end. So whatever you have left goes at the beginning.
Okay.
Okay, so if you're on a 26-day cycle, then you're going to ovulate on day 12. Right? 26 minus 14 is 12, and so that's the day that you ovulate on.
Okay, thank you.
All right. Okay, so so the luteal phase then is that ruptured follicle. It's going to collapse to form the corpus luteum. The endocrine gland secretes progesterone and estrogen, begins to degenerate in about 10 days, and then the corpus albicans remains. When you get your charts on for Monday, then we'll just go step through the chart and it'll put all of this information all together, especially the graph that comes on the back of it. It really is nice to see how everything lines up. Okay, so here's a ovulating oocyte. I don't know how we get pictures like this, but here is the wall of the ovary right here, and then here's the fluid from the antrum, and there's the oocyte itself. Really cool. All right, so let's get into all the funky hormone stuff that's going on in the female, and then we'll probably wrap it up for today. All right, so estrogen is a great inhibitor of GnRH and FSH and LH. Okay, so the little, well, not the ovary, not the baby's ovary, but the adrenal cortex. And to think, what gland? So the adrenal cortex is going to be producing androgens, which will then get converted to estrogen, and that will inhibit the hypothalamus until puberty. And then something magical happens. I don't know. The hypothalamus becomes insensitive to that estrogen inhibition, but not permanently. It just so that you can start cyclically having a period. So somewhere in puberty, and it's different for every girl, she's going to have her first period. We call that menarche, and yay, you had your first period. Okay, always remember, how old were you when you had your first period? Oh yeah, I still remember that day. What am I? How old am I? 42 years later, something like that. Anyway, okay, so hormonally then, as we go through this 28-day cycle, what's being secreted? So day one, which is when we started the follicular phase, when we were having our period, okay, so we were cleaning up the room from the last lack of tenant. And GnRH, gonadotropin releasing hormone from the hypothalamus, will stimulate the release of FSH and LH from the anterior pituitary. So then FSH and LH will then go down to the ovary and stimulate follicle growth and the thecal cells. As the follicle grows, estrogen secretion begins from the thecal cells, and then androgens are released to be converted to estrogen from your adrenal gland. All right, then we start to get, and I'm just going to show you a little picture of how this works. So the negative feedback of estrogen on FSH and LH release occurs. Positive feedback on estrogen production. And then we also get another hormone called inhibin, which also inhibits the releases of FSH. So let's go to that now and see what's going on day-wise. Okay, so day one, step one, GnRH is going down to the anterior pituitary and stimulating this first. Okay, because here's a second one, but we want to have this first secretion of FSH and LH on the follicles. So here the follicles are growing. So then we get here to this well-developed primary follicle with the thecal cells and the granulosa cells and the thecal cells around the outside. This estrogen that it's producing right here, so slightly elevated estrogen and rising inhibin levels, are going to come back and inhibit the release right here on the anterior pituitary. So I still have GnRH is still going down to stimulate the production of FSH, but I'm not releasing FSH and LH. Okay, so that's an important difference. Not that I'm inhibiting its production, but I'm inhibiting its release. Okay, so then we're going to go down to about day 12, day 11, day 12. Now I have such high estrogen levels at this point that now, oh look, I have a stimulatory effect on the anterior pituitary. And so now what we're going to do is release FSH and LH in a large number. In fact, the LH is factors of, like 100 times more what we were secreting before. And so we get this, where is it? With LH surge, and then we get a quite a bit lower FSH surge, but it's the LH surge. So I get this spike in LH secretion on about day 13. So then day 13, if we're going on the 28-day cycle. Okay, so the day before ovulation. So then this is going to come over here. The LH is going to come over here at this step, act on the mature follicle and stimulate ovulation. Now, the LH is still high. Now we're going to be on day 15. So here we're day 14. So day 13, day 14, day 15, and we're going to keep stimulating the corpus luteum But the progesterone and estrogen that are produced by the corpus luteum now come back and inhibit both the GnRH and anything going on in the anterior pituitary, because now I have to, I've advertised for a tenant, and now I have one that says it'll move in, but only under the condition of getting a roommate or, you know, being fertilized. So I wait 2 weeks to see if this ovum gets fertilized. So in the meantime, I can't advertise for another tenant. I have to wait to make sure. I have to give this one its fair chance to be able to move into my room, okay, into the uterus. So I wait for 2 weeks. So I stop any advertisement from going on at all. Now, this LH surge is now going to decrease. And so as these levels are declining, because it's going to still stimulate the corpus luteum, but as these LH levels decline, the corpus luteum is going to go, oh, I'm not getting any signal that I need to wait, and my tenant didn't tell me that they moved in yet. And the tenant, if it gets fertilized, is going to secrete HCG, human chorionic gonadotropin. And that will be the signal to the corpus luteum that somebody moved into the room. And then it won't get rid of it until the baby leaves, either through a miscarriage or an abortion or birth. Okay, so so that's this negative feedback. And this is powerful negative feedback. This is why we have birth control pills. So this is what's in birth control pills, usually estrogen, but it could be progesterone or a combination. And this is what's stopping the secretion of GnRH, production and secretion of GnRH. Okay, so let's just go back and look at the notes, and then we'll be done for today, because like I said, I really want you to have your chart. All right, so high estrogen levels exert now a positive feedback on the anterior pituitary, day 12. Day 13, about 24 hours prior to ovulation, an LH surge occurs, and then to a lesser extent FSH, and then we get that. Okay, and we'll just finish this off. So it says the LH surge stimulates the primary oocyte of the dominant follicle to complete meiosis 1. Secondary oocyte continues to metaphase 2, where it will sit until it gets fertilized. And then LH also triggers ovulation about day 14. So day 14 is the ovulation day. Estrogen levels decline, corpus luteum forms, and then increasing progesterone and estrogen form or from the corpus luteum now have an inhibitory effect again on FSH-LH. So here's that little chart that I was telling you about, how to match everything up, but I have some more stuff on our chart. I also have the events of the ovarian cycle and the events of the uterine cycle stuck in with this as well, so you can line everything up and go day by day what's going to happen. So I'm just going to leave you there with that, and then make sure that you have the thing on Monday. And so I don't want to go any further today, because we can get confused and lost with events and days and hormones and all sorts of stuff like that. So like I said, it's confusing. And so here's my last little story. Excuse me. So I had this student, bless his heart, sitting in the front row, and so we're going through all of this, and I had, for a couple of lecture days, I had talked about, you know, all the events of it, just the women's cycle. And so he raises his hand, he goes, Ms. G, when are we going to have the baby? And I said, Paul, we haven't even had sex yet. I mean, there was no fertilization. He thought in all of these steps that were going on that soon a baby would be born, and that poor ovum hadn't even been fertilized yet. So he was just, he was so lost. Like, I don't even know what we're doing. So it was about that time that I came up with the chart, and so that's helped everybody put things together a little better. So we'll make sure we have that on Monday. And other than that, I don't have anything more for you. Good luck on taking your test. It keeps popping up on my phone that you guys are taking your exam, so that's good. Don't forget about your lab practical. Is due on Tuesday, so get that done, because I'm not opening up late stuff. You guys, you can stay on things now. We're settled down into a routine well enough that I think you guys can keep track of everything. So just follow through the modules for what stuff is due when, and we should be good. So I hope you have a great weekend. I'm looking forward to, after I take my test this afternoon, to having a fabulous weekend, because for the first time in a long time, I don't really have anything to do. I can clean my house. Woohoo, I'm excited to clean my house. All right. Okay, so I guess that's all. You guys have a